So to start off, we live in an age marked by a heightened interest in space exploration. But with this comes a growing need to investigate the challenges space travel poses to astronaut health. Of particular concern, astronauts in space undergo physiological changes reminiscent of aging. But what if I told you that there's more to that story? Because from a cellular level, space travel could really be the fountain of youth. And the reason why involves some things called telomeres. Telomeres are the repetitive DNA that cap the ends of our chromosomes. You can sort of think of them as the caps on your shoelaces. Without them, our DNA would get frayed. Something important to know, though, is that telomeres don't last forever. As cells age, their telomeres shorten. So because of this, telomeres have been seen as biomarkers of our overall health. Longer telomeres are linked to youth and vigor, while shorter ones are linked to age-related illnesses, and even some cancers. As previously referenced, astronauts in space experience degeneration that seems to mimic aging. As a result, their telomeres are expected to shorten faster. However, astronaut telomeres have been found to lengthen aboard the ISS and shorten only after the return to Earth. So, like I had said, this could suggest that astronauts in space get more youthful on a cellular level. But we're not completely sure about this because we don't know how or why this process even happens. So, in my experiment, I plan to investigate a potential cause of these spaceflight telomere dynamics. Stem cells. Now, why stem cells? Well, they have the longest telomeres in the body, and upon differentiation, they'd initially maintain this trait. Further, most stem cells are involved with healing. Something that I think should trigger healing in astronauts is cosmic radiation. After all, radiation kills cells, and space is full of it. So I think that in astronauts, radiation-induced cell death reaches a point where it stimulates stem cell activity. And then the result of this activity would be an influx of younger cells with inherently longer telomeres that could then cause the overall cell population to have longer telomeres on average. And back on Earth, where radiation is decreased, it makes sense that decreased stem cell activity would follow. And that would mean that more cells would be getting to live longer and develop shorter telomeres as they age. And this thinking isn't purely hypothetical. A study linked oxidative stress during spaceflight to these telomere dynamics. And that same study also noticed increased telomerase activity in astronauts immediately post-flight. And as some background, telomerase is an enzyme active only in stem cells. Lastly, astronauts tend to have more white blood cells in space, which suggests that they have increased stem cell activity in space. So, in order to investigate this, I would be using adult male Caenorhabditis elegans. They're small, easy to raise, and they have conserved biology with humans. And also, they challenge my theory in a pretty interesting way. You see, the only stem cells in the adult worms are their reproductive germ cells. But these cells aren't known to help with healing. So it was pretty perplexing to learn that C. elegans was found to develop slightly longer telomeres after 11 days in space. But while this may seem conflicting, the authors of that study stressed that their results were not conclusive because of the inconsistencies between the maintenance of the spaceflight samples and their ground controls. So this leaves it entirely possible that the telomere lengthening observed may not have been due to spaceflight. So with that, I can get into the purpose of my experiment. The goal is to see whether C. elegans actually develops longer telomeres, despite its lack of healing adult stem cells. Investigating this question would help us figure out if the proliferation of healing adult stem cells is what drives astronaut telomere dynamics. So in my experiment, I hypothesized that C. elegans will not develop longer telomeres in space, regardless of whether it has its germ cells or not, for the reasons stated earlier. So this leads me into my freeze and fly. It's a way for us to make sure that we can accurately measure telomeres in space. So for this experiment, 16 adult male worms will be lysed and then put into tubes of PCR master mix. Half of these tubes will then be sent to space. But in both space and Earth, each of the categories here will be represented by 2 worms. The first category is for standard C. elegans. The second category is for a strain of C. elegans with telomeres naturally longer than those of the standard worms. The third category is for dead worms, while the last category is for worms that had their germ cells laser ablated with ease. By including the extra strain as a positive control for longer telomeres, we'd be able to make sure that we could differentiate between different telomere lengths in space. And dead and sterilized worms will be used in my ambitious experiment, but I've included them here so that we can make sure that their modifications wouldn't affect their telomere readings in space. So in both conditions, telomere lengths will be measured using a PCR-based approach called STELLA. This approach amplifies the telomere of a specific chromosome in the C. elegans genome. After this step, telomere fragments will be marked using cyber-green probes and then viewed with a fluorescence viewer. The longer the telomere, the brighter the fluorescence. And as shown, I think we'll get accurate PCR results in space. Now we can move on to my ambitious experiment. Here, we'll really get to test my hypothesis. This time, we'll only be using standard, dead, and sterilized worms. And unlike the freeze and fly, each set of animals would spend 11 days in their respective conditions. So with that, the dead worms will become the negative controls for telomere elongation. And this would leave us with the standard worms as the primary experimental group, while the sterilized worms would be used to isolate the effects of germ cells on any observed telomere dynamics. After the STELLA PCR, the results of the space worms will be compared to those of their corresponding ground controls. If both the standard and the sterilized worms develop longer telomeres, that is strong evidence that stem cells don't drive their telomere dynamics. But if neither the standard nor the sterilized worms develop longer telomeres, that supports my hypothesis and suggests that healing adult stem cells are needed for spaceflight telomere dynamics. So if given the privilege of being conducted aboard the ISS, this experiment would help us figure out how spaceflight telomere dynamics works or doesn't work. But more than that, it would take us one step closer to figuring out the long-term effects of longer telomeres on astronaut health, such as to see if it actually makes astronauts more youthful or maybe lowers their risk for one illness, but raises it for another. And these questions are even more important to ask as things like commercial spaceflight and long-term Mars missions are being explored. And further, if stem cells were found to drive astronaut telomere dynamics, maybe that could be applied to medicine on Earth to counter the effects of age-related illnesses, treat short telomere syndromes, or even prevent certain cancers. But even if my hypothesis were not supported, the findings would still narrow down the causes behind this phenomenon so that future research could still lead to these exciting outcomes. So although telomeres cap the ends of our chromosomes, in my experiment, they'll be used to uncap the mysteries of human adaptability and show us if longevity truly stems from spaceflight. I'd like to give thanks to my mentor, Ana Carla, my sponsor, Ms. Manning, the entire Genes in Space team, as well as the Genes in Space corporate sponsors.